Hi, my name is Annie, and I'm a summer co-op student at the University of Guelph Crop Science Building. Today we're going to be doing PCR, better known as polymerase chain reaction. Before starting, it is very important to be safe within the lab. That's why you must always have gloves, lab coat, safety shoes, and safety glasses as well. So the tools I'll be using today are the calculator for calculating the formula, pipette tips, a pen for labeling, as well as pipettes. And in our safety video it says not to pipette with your mouth, so remember not to do that. I have already labeled my PCR tubes and my master mixes tubes so that I do not confuse them. So now for the fun part, the pipetting. So your goal is to get all these chemicals into one tube with the correct amount and then put them in the PCR tubes. So now I've put all the chemicals in the master mix tube, and now it's for centrifuging. So now that I have my centrifuged master mix, I'm going to fill them in the PCR tubes and then add DNA in them right after. Now we are at the centrifuge machine. This machine simply spins the materials, thereby increasing the G-force inside the tube and ensuring that the liquids on the sides of the tubes are forced down into the bottom and that there are no bubbles in the solution. So now we are going to put the PCR tubes into the centrifuge machine. And you must remember to always balance. So sometimes when you put a small amount of liquid into a centrifuge, a little pocket of air gets trapped, and this affects the transfer of heat to the solution during the time that the tubes are in the PCR machine, causing results to be inaccurate. So we are going to avoid that problem today by using the proper techniques. Now that we're finished at the centrifuge machine, we're at the PCR machine over here, and what this does is that through a series of cooling and heating, it amplifies the DNA. So now that all the PCR tubes are in the PCR machine, we're going to run the program. And stay tuned for electrophoresis.